In this section, I'm going to talk about isolation, inspection, and identification. So let's start with isolation. A pure culture is a medium that contains only a single known species or type of microorganism. It's used most frequently for laboratory studies. Anexic is free of other living things except for the one being studied, and a subculture is a second level culture from a well isolated colony. So, just kind of walking you through the process of if we were to culture microorganisms in a laboratory. Those microorganisms are going to have to come from somewhere. So, usually, when we want to prep for one of our academic labs, our lab tech is going to get on the phone with a biotech company and say, I need a pure culture of Staphylococcus aureus for my lab. And then they'll ship a pure culture to us. Now, that pure culture sometimes comes lyophilized or freeze dried. Sometimes it comes on a slant, so it's a living organism, but it's been isolated from original source, so that that culture is a pure culture that's coming directly from a source. Well, from there, our lab tech will then subculture it. So we'll take that original culture, we will reactivate it, so whether it needs to be mixed with solution and kind of woken up a little bit, or whether it's ready to go from a slant, we're going to then culture it into a second media source. It's still a pure culture, we're just taking that original culture and subculturing it, we're putting it into a new media. And then we're going to use it in experimentation. Mixed and contaminated cultures. When we're talking about patient specimens, most patient specimens are going to be mixed cultures, and some are going to be contaminated. So if you think about it, let's take a nasal swab from a patient. Well, that nasal swab has tons of different types of microorganisms. It's never going to be that one organism that we're looking for. So that's a mixed culture. Places on the body where you might have a pure culture would be sites that are normally sterile. So cerebrospinal fluid, that's supposed to be sterile. There's not supposed to be microbes growing in there. But if a patient develops meningitis or inflammation of the meninges surrounding the brain and spinal cord, they're going to have symptoms, and you're going to do a spinal tap, and that spinal tap is then going to be evaluated for the presence of microorganisms. Typically, only one species is going to be in there. So that's going to be a pure culture, in essence, from that location. But with those rare exceptions, most of what we're going to get from a patient are going to be mixed cultures. So mixed culture contains two or more identified, easily differentiated species of microorganisms. In a contaminated culture, a culture that was once pure or mixed now contains contaminants or unwanted microbes of uncertain identity. So contamination is unintended growth or unwanted growth. If we were to take that spinal sample and the needle was not cleaned prior to using it, which would be absolutely terrible, or if the needle, when drawn out, happened to also draw in some mold spores from the environment, and we let that culture sit for a while, that's going to eventually produce that mold organism that was in there originally, especially if it's incubated at room temperature without the presence of light. You're going to see mold start to grow from there. And that mold is going to be contamination. It was a pure culture, and now there's contaminants. Here's a picture of a pure culture. So here on solid medium, you can see only one species growing in each one of these. And this one is a mixed culture. You can see on the plate there's different colored species. So there's some tan ones and some yellow ones. Contaminated, you can see this pure culture all of a sudden has this unwanted growth or this unintended growth on the plate. In order to isolate or separate out species in a mixed culture or even in a contaminated culture, we need to use particular techniques. So a colony is a discrete mound of cells that's formed on a solid nutrient surface, and it consists of just one species and no other if it's formed from a single cell. Isolation requires a small number of cells, a firm surface, a petri dish, and inoculating tubes or tools like loops. So I'm only going to discuss the streak plate method because this is the method that we're going to learn about during the lab portion of this course. So in the streak plate method, a small droplet of culture or sample is spread across the surface of a medium with an inoculating loop. The pattern used to inoculate gradually thins out the sample and separates the cells in order to encourage the growth of discrete colonies. So basically what you're going to do is you're going to put bacteria on your loop. Now you want to flame sterilize your loop before touching the bacteria, and then you're going to attain a small amount of bacteria, and then you're going to open up the plate just slightly so that you can get into the plate, and then you're going to spread bacteria around in one region of the plate. Then you're going to flame sterilize your loop again. Flame sterilizing your loop will kill any bacteria that's on the loop. You allow that loop to cool, and then you're going to take some bacteria from the first region that you spread it on the plate and rub it into a second region. The goal here is to rub the bacteria thinner and thinner and thinner until you have isolated colonies. So, when you rub the bacteria in the first place, it's really concentrated. But when you're moving a small amount from the first to the second region, you are reducing the number of microbes that are on that region of the plate. So then you're going to take your lube and you're going to flame sterilize it again, cool it off, and now you're going to rub the bacteria from the second region to a third region. And just a small amount of bacteria again to rub it thinner and thinner and thinner. And then you're going to flame your loop again, let it cool, and then rub it from the third to the fourth region. By the time you get to the fourth region, there should be isolated cells landing on the plate. You won't be able to see them, but after incubation, this is what it's going to look like. You're going to see a lot of growth in one region, less growth in the next region, even less growth in the next region, and then finally you have isolated colonies. The reason for isolating colonies is so that we can identify the species within a mixed culture. If we see a mixed culture has hundreds of one type of bacteria and then a few of other types of bacteria, the one that's usually the pathogen is going to be the one that's most numerous, or it's going to be the one that causes things like beta hemolysis, really bad hemolysis. We can start to examine what the bacteria on the surface look like to try to figure out which one is the pathogen and which one is the normal biota, the normal natural bacteria that's in the background. So isolating allows us to identify and diagnose a patient's infection. Now I'm going to speak briefly about inspection and identification. So during inspection, you're going to run various tests on a microorganism in order to identify that microorganism or assign it to a genus and species. Microbial profiles are determined through combining phenotypic testing, genotypic testing, immunological, microscopic and macroscopic testing. So I'm going to give definitions to each one of these now. Phenotypic testing. Phenotype is an appearance of something. So in phenotypic testing, we are examining the appearance of the microorganism. Examples of phenotypic testing would be doing a gram stain or identifying colony morphology. Colony morphology has to do with the size, shape, and color of the colony, and that can help us with diagnostics. Genotypic testing. Genotypic testing has to do with evaluating the genetic material of the organism. We can do this in a variety of different ways. We can sequence a portion of the genome. We can use special DNA probes to find certain sequences in the organism. Or we can try to use a process called PCR, polymerase chain reaction, to amplify a particular region of the genome to see if that gene is present, and then that can help us in diagnosis. Genotypic testing is going to be talked about further when we get to genetics. Immunologic testing, that's using the antibodies in order to make a diagnosis. We can do this in a couple ways. We can either have antibodies against a known pathogen and then expose that pathogen to those antibodies to see if that pathogen is what we know it to be. Or we can look for the presence of antibodies in the patient's serum. If there's antibodies in the serum, then we can pretty much assume that that patient has been exposed. We don't know if it's a current or active infection, we just know that they've been exposed. Macroscopic analysis, that's something that you can see with the naked eye, while microscopic analysis is something that you would need a microscope for. As we go throughout the course, we're going to be talking about different types of testing and different ways that we can diagnose infections. So I'm giving you these definitions up front, and then we'll talk more about them as we go throughout the course. There's biochemical and other types of tests. So biochemical tests can determine fundamental chemical characteristics, such as nutrient requirements, products that are given off during growth, the presence of particular enzymes, and their mechanisms for deriving energy. In the next section, in the next set of lecture series, we're going to be talking about biochemical tests in detail. Other analytic and diagnostic tools include genotypic testing and immunologic testing. It detects microbes based on their DNA, and immunological testing tests the isolate against known antibodies.